Okay, so let's get started. So when we talk about the digestive system, there's 5 main functions that we want to be able to talk about when we're finished this particular topic. So one thing is the function of ingestion, which is about introducing food to the digestive system. Then we have to be able to break up that food into small enough components that we can utilize them. That's digestion, and digestion also involves some secretions. And then if we're successful with those things, we're going to absorb the nutrients into the system, and we're going to get rid of whatever is not very helpful. So if we look at the components of the digestive system, we start with the same structures as we did for the respiratory system. In other words, we've got an oral pharynx, we've got a tongue, we've got a hard palate. So there's a nasopharynx and oral pharynx, the pharynx in general. And in this case, when we're following the digestive system, we want the pharynx to run into the esophagus. And I'm actually going to change the color of the esophagus. And the esophagus is a tube that's going to run into the stomach. And the stomach is a J-shaped receiving pouch. And then we're going to run from the stomach into the small intestine. And the small intestine is a series of coils that's going to run into the large intestine. And the large intestine has an ascending component, a transverse component, a descending component, and we finish at the rectum. Now, clear up some of this just so it's not totally confusing what we're looking at here. So the point of me changing the colors as we went through is to indicate a change in appearance or a change in form, structure, is going to represent a change in function. And this is a recurring theme throughout the organization of the intestinal system, of the gastrointestinal system. So we can appreciate here with what we've talked about, with what we've shown, is that we've got food that might be introduced to the mouth. In other words, ingestion. And that's going to involve the process of swallowing. And that's what introduces the food to the digestive system. Now, the goal of the digestive system is going to go through the rest of those functions that we listed on the left-hand side of this page. But the idea is that as long as the food is in this tube, no matter where in this tube it is, it's technically outside of the body. In other words, it's of no use to us if it stays within this tube. This tube is technically external. Here we have an opening to the external environment, and down here we have an opening to the external environment. And as long as the food stays within that tube, it's technically external. So the purpose of digestion, and we're going to use mechanical means and secretory means, in other words, chemical means by introducing enzymes, we need to get absorption. And absorption by definition means we're going to take it from that tube and introduce it to the circulatory system. So that's the goal of the gastrointestinal system, is to get those food particles broken down small enough so they can be absorbed and utilized. So think about amino acids. They come from proteins. Proteins come from meat, for example. So you eat a chunk of meat, and the goal of digestion is to break them down into small enough components, usually they're amino acids or small peptide chains, so that they can be absorbed into the circulatory system and rebuilt into human proteins. So you ate an animal protein, and you're going to rebuild into human proteins from the building blocks. So, and the same concept would apply for lipids and carbohydrates. So that's our goal. Now, when we look at this tube, it's basically a muscular tube. Most of the muscular tube is going to be under involuntary regulation. In other words, autonomic regulation, or sometimes some textbooks like to refer to an enteric nervous system, enteric referring to intestines, and in this case, the digestive system. So it's a category really of the autonomic nervous system. One of the major nerves involved here would be the vagus nerve. And remember, parasympathetic division was about rest and digest. So this is the system that we the part of the nervous system that we're concerned with when we look at influencing the rate of secretions and motility through that digestive system. So the first thing to recognize with respect to the typical organization of this tube is that we're going to have a lumen. So a lumen is the opening of the tube, and that lumen is going to have a set of single or simple columnar epithelial cells. So our innermost lining is called the mucosa, and the mucosa is made up typically of a simple columnar epithelium. That simple columnar epithelium is going to have some columnar cells that produce some mucus and some that are going to have other functions as we move down through the system. But it's called a mucosa, so mucus is right in the name. And then the next layer that we have is called the submucosa. So this next layer is a submucosa. We're going to find some glands, and the glands are formed by invaginations. So in other words, we turn in, and that would be a mucosal gland. If the gland was formed within the submucosa, they would be submucosal glands. And in the case of the submucosal gland, the gland would be found in this layer, but the duct would empty into the lumen. So we would secrete into the lumen. And we also, I'll show you a nicer picture of this in just a minute. We also have a nerve supply associated with this, this layer, the submucosal layer, and that's going to be responsible for influencing the rate of secretions produced by these glands found in the mucosal and submucosal layers. Then we're going to have the next layer is a muscularis layer. And the muscularis layer is going to be a layer typically of two orientations of smooth muscle. There's a circular orientation and there's a longitudinal orientation. So two layers of smooth muscle. Circular. So when the circular layer contracts, what's going to happen to the lumen?
Constrict.
That's right. It's going to, the lumen diameter is going to be smaller. It's going to get smaller. And what would happen when the longitudinal layer of the smooth muscle contracts? Well, if it contracts,
Want to move the food like in the direction, like to the center?
it's going to shorten, right?
Yeah.
Longitudinal, it's going to shorten. So what we're going to see with the circular layer is constriction of the diameter, and then with the longitudinal layer, a shortening of the length, that particular length of the tube. And when those two contractions work in a sequence, we get what we call peristalsis. And peristalsis is going to propel the food along the length of the tube. So that's going to start at one part of the tube, and it's going to continue throughout the length of the tube. And we're going to see different areas where we can slow it down or speed it up, depending on the conditions and the circumstances. And all of that is going to be surrounded by an outermost layer called the serosa. So that's usually going to be a loose type of connective tissue that's well vascularized. It's going to be where the blood vessels are located that are going to supply these tissues. We've got a nerve supply that runs between the two muscular layers that's going to be responsible for contraction and relaxation cycles, in addition to the fact that this is smooth muscle. So there's going to be some local factors that can influence contraction. And by local factors, I mean changes to the diameter of the tube because of the mass of food. So when the food enters, for example, the esophagus, and let's go back to our original color here. So if we create a mass of food in the esophagus, and the esophagus is stretched, so our stretch has happened on either side, that stretch initiates a response from the smooth muscle, contraction. So that's where peristalsis would get started. And it would start on a little bit of a delay, so it's going to contract slightly behind the mass of food. So when we constrict behind the food and then shorten the length of the tube, the result is the food is going to be pushed down the length of that smooth muscle tube. Did you follow me okay with that? Does that make sense? Yes? No?
Yeah.
Okay. Yeah. Somebody's participating. All right, so let's take a closer look at this same organization that we're talking about here. So here's a little fancier picture of the same thing. This represents the lumen. Okay, and so this first layer of cells that we can see as a thin sort of beigey colored line in there is our mucosa. So here's our mucosa, and it's made up of an epithelium, that's the simple columnar cells, and a lamina propria, which is a fancy word for basement membrane. We know that our epithelial tissues have to have a basement membrane because that's the transfer between a source of blood, which in this case is going to come all the way from the muscularis layer, okay, down into the epithelial tissues through absorption. So this represents our simple columnar epithelium. This is a mucosal gland right here, and this would be a submucosal gland right there. Here's our submucosal plexus of nerves that are going to influence secretion rates of glands, okay? And then when we look at the muscularis layer, circular is going around, so it will be involved with reducing or relaxing the diameter of the muscular tube, and then the longitudinal muscle, okay, will shorten the length of the tube. The nerve plexus, remember plexus is just a fancy word for a bunch of nerves that branch off and distribute, is myenteric. So myenteric gives you a pretty good clue that it's going to be associated with muscle tissue. So we have the two sort of networks of nerves, one that's going to influence secretions and one that's going to influence muscle contraction in the tissue. And that would be primarily parasympathetic divisions. We have some sympathetic, but we know that sympathetic will cause relaxation and slow down secretions and motility, whereas parasympathetic is going to increase motility and increase secretions. Okay, so this is the general organization of the tube. Now, what I want to focus on before we start to talk about individual sections is I color-coded our digestive tube. Everywhere there's a change in color, there's going to be a slight change in the structure, sometimes a major change in the structure. And the point of that is to recognize that if we change structure, what else is going to change?
Function.
Yeah, function. So the point of this is, and this is a good thing for you to remember for at least a week or so, is that if we change the structure, we're going to change the function. So you can look at this and you can look at that blue tube that we're going to find out is called the, I better label some of this. So this is the esophagus. And the esophagus is a muscular tube that's responsible for delivering the food from the mouth through the pharynx to the stomach. And in the stomach, you can see the green part is the stomach. So the stomach is an expansion of that tube. So it looks very different. And that different appearance is going to result in some different functions. And from the stomach, we enter the small intestine. And then from the small intestine, we're going to get to the large intestine. We finish down here at the rectum, and we're going to eliminate from the rectum. So we've got different structural features that represent different functional responsibilities as we move along this muscular tube. Does that make sense? Everybody should be comfortable with that concept by now.
Yeah.
All right, so let's start with the mouth. Now, the mouth, we can appreciate, has teeth. It has a tongue. So already we've got several exceptions to the typical arrangement of the digestive system. And we should be okay with that because we also recognize that the mouth shares responsibilities for example, breathing and for speaking and that sort of thing. So we expect to see some differences here because this is not an area that's dedicated just to the digestive system. But instead of mucosal glands, we refer to those glands as salivary glands. And we have 3 dedicated salivary glands, pairs of salivary glands. The largest of those is called the parotid. And those run just in front of your ear down to the angle of the jaw. So they're the parotid glands. And then along the length of the jaw are the submandibular glands. So if you run along the angle of your jaw towards your chin and just go inside that bone into that soft area there, that's where you'd find the submandibular salivary glands. And then underneath the tongue, so sublingual, we've got the salivary glands. Or sorry, underneath the tongue, we've got the sublingual glands. Now there's a 4th pair that's a bit controversial. So it's controversial for 2 reasons. One reason is that it had been ignored or missed historically by anatomists and physiologists for decades. So just in the last decade or so, a group of scientists discovered what they considered to be the 4th pair of parotid glands, and they're associated with the auditory canal. You remember the auditory canal drained the middle part of the ear into the nasal pharynx and is a common site for the potential for earaches and inflammation of that middle part of the ear, especially in young people. But that 4th pair are called tubarial because they're associated with the eustachian or the auditory tube. So that's where the name came from. But it's controversial because they were found on the distal end of that auditory tube, just around the range where it empties into the nasopharynx. And the nasopharynx, most of the time, is considered part of the respiratory system and not part of the digestive system. So in that respect, a lot of sort of scientists and anatomists don't consider these to be salivary glands because they don't directly contribute to digestion. They lubricate the airway, especially around that auditory canal and the nasopharynx area, but aren't really directly involved with digestion. So that's more of an interest than it is a requirement in terms of memorization or anything like that. The ones we know are directly associated with the digestive process are the first three pairs that we've listed there. The other consideration that makes the mouth unique and different from the typical arrangement of the muscular tube is that the muscles up in this mouth are skeletal muscles, which means they're under conscious control. So skeletal muscles for the tongue, skeletal muscles for the cheeks, skeletal muscles for the first part of the pharynx, and even the very first part of the esophagus are all under conscious control. And what that allows us to recognize is that the first part of the digestive process is going to be a voluntary process. And that doesn't come as a great surprise to anybody. We choose to eat. We choose the foods we eat, and so those are all conscious decisions. The other thing to realize is that we've got taste receptors. And this helps us with our choices that we make to eat. So we know generally about taste receptors. We talked about those in AMP 100. But taste receptors come in four main categories from the perspective of taste. And that's salty and sweet, which tend to be favorable and sort of hardwire us to encourage us to swallow. And then we got bitter and sour, which are sort of the opposite of that. Those are sort of hardwired to give us a warning that there's a potential for toxicity in the food source. And we can, of course, overcome that by voluntarily choosing to eat something that's sour or bitter and that kind of thing. And yes, there is the umami taste receptor, and there's some people believe that there's receptors for water, and they throw those in to this category as well. But the concept is based on the fact that these taste receptors are going to be found on the tongue and on the back of the pharynx, and together with influences from the olfactory senses, we know that olfactory sensation helps augment the taste sensation. In other words, if the olfactory sensors are not working effectively, your taste receptors are not quite as sensitive. Most of you know that if you've got sinuses that are blocked up and you're not smelling well during a cold, for example, your food tastes all kind of the same or bland. And that's because we've lost that contribution of the sense of smell that contributes to the sense of taste. So when we look at these salivary glands, they produce anywhere from 500 mils to maybe 1.5. I guess I should stay consistent here. 1,500 mils per day of secretions. And most of that's water, plus some electrolytes. And we've got also some enzymes. So there's an enzyme category that's called salivary amylase. And if you see that word amylase, ASE tells you it's an enzyme, so we're okay with that. And the amylo part tells us it's going to target carbohydrates. So what this represents when we talk about digestion is a chemical type of digestion, because enzymes are acting on the chunk of bread or the pasta or whatever the large molecule of carbohydrate happens to be. When the teeth grind the food and the tongue mixes it with the saliva, that's mechanical digestion. So all the way through this system, we'll be talking about areas where either mechanical or chemical digestion is the focus. But the goal in both cases for digestion is to break down large molecules small enough that they can be absorbed. So there's also a lingual lipase enzyme. Now, if it's called lipase, what food group do you think it's going to act on?
Lipids.
Yeah, lipids. It's not a big leap to figure that one out. But the thing about the lingual lipase is it's inactive at the mouth. So this becomes kind of a key thing to keep in mind. If I was to ask you a question, for example, about where the first location is for chemical digestion of lipids, it might be easy to say, well, lingual lipase. Lingual has something to do with the tongue. It's released by salivary glands. The first location for chemical digestion of lipids would be in the mouth. But lingual lipase doesn't become active until it's in an acidic environment, and most of the saliva is alkaline. So the lingual lipase won't be activated until it travels to the stomach. We'll find out in just a few minutes how acidic the stomach is, but the short answer is it's very acidic. So lingual lipase is going to be produced by saliva, but not active until it reaches the stomach. So that's a key thing to remember about lingual lipase. So mechanical digestion begins in the mouth for everything that's found in the mouth because of teeth and the tongue and mixing with the saliva. So the other thing to keep in mind about saliva is that it also has the responsibilities. You can appreciate that it's going to moisten food. Obviously, it's going to expose food to enzymes, but the enzymes that are targeted in the mouth are primarily going to be working on carbohydrates. It moistens food, which also increases taste sensation. So in other words, when the tongue is moist from saliva, we get better sensation for taste. Increases taste. I'm just going to put sense in there. Because of that fluid component, it's going to aid in speech. Most of us can appreciate that when we have saliva in our mouth, we speak better. It's not as sticky, literally. There are some additional enzymes, antimicrobial. Because as you can appreciate, there's going to be a lot of potential for microbes to be introduced to our system through our food. Our food is grown in soils, our food is exposed to air, and there's microbes floating in the air, and there's microbes growing in the ground. And so there's a big likelihood that we're going to have components of those microbes introduced to our through our digestive system to our bodies. So we have some antimicrobial components, and the estimates are anywhere from 8 to 500 million bacterial cells per milliliter of saliva. So saliva is exposed between what grows naturally in our mouth and what's introduced through our food, anywhere from 8 million to 500 million bacterial cells per milliliter of that salivary composition. So pretty significant. And for most of us, we don't get sick. But it is these bacterial cells that are metabolically active that can be the source of bad breath. Just like we know that they can be the source of body odor in our armpits and groin areas because of their constant feeding on lipids and proteins and carbs that we provide for them through our sweat and through our sebum. So there's some other components to the saliva, but basically, we say that it's slightly alkaline, and so that's why that lingual lipase won't be active there. So to get this stuff, I'm going to introduce a couple of new layers here. Oops. To get food delivered from the mouth to the esophagus, the next component we have to swallow, which is also known as deglutition. So the fancy word for swallowing is deglutition. And we have a voluntary component and an involuntary component. So voluntary involves placing the food on the tongue and in the mouth, and then we're going to choose through conscious effort, conscious signaling to the muscles of the tongue, muscles of the mouth, and cheeks to push the food to the back of the pharynx. And from the pharynx, we're going to direct the food towards the esophagus. Okay, in the involuntary phase, voluntary is conscious mixing and delivery to the esophagus. Involuntary, there's two distinct phases to the involuntary phase. We got a pharyngeal. In the pharyngeal phase, that's the component of delivery from the pharynx to the esophagus. Okay, so we're going to have muscle contraction. There's my square brackets for contraction. Muscle contraction of pharyngeal muscles to the esophagus. So what we can do is, just to clarify here, let's just put this to the pharynx, just so we don't get mixed up. So the pharyngeal muscle is going to contract to deliver specifically into the esophagus, and they contract to lift glottis, remember, from the respiratory system towards the epiglottis to prevent food from entering the airway. So that's really what the pharyngeal muscles are responsible for in this late phase of swallowing. And then we have the esophageal phase. The esophageal phase is going to introduce peristalsis. So that was that involuntary sequence of contractions that's going to result in propelling the food further along the digestive tract. Okay, so that brings us to the esophagus. And with the esophagus, it's basically a muscular tube. So we're going to see in the esophagus the first level of organization that represents those typical arrangements: mucosa, submucosa, smooth muscle layers, and the serosa around the outside. The one difference is that this muscular tube is somewhere between 20 and 25 centimeters in length. The first third or so is still skeletal muscle. And I didn't mention for the mouth, but with the mouth, it's a stratified squamous epithelium. So again, we're not going to see that simple columnar epithelium that we typically see. In the mouth, it's subject to more friction from dry foods or large molecules of food. So we're going to see a stratified squamous epithelium. And in the proximal third or so, we still see a bit of that stratified squamous epithelium. So then somewhere along the middle section of the esophagus, we are going to switch to smooth muscle and the simple columnar epithelium. Now, the other thing to keep in mind is from a functional point of view, the sole responsibilities of the esophagus is the delivery of food from the mouth to the stomach. Okay, so basically no digestion is going to occur in the esophagus. The esophagus is just a delivery system. So once we've chosen to introduce food to the back of the pharynx and then the esophagus, it's peristalsis to the stomach. Few seconds between the mouth and the stomach. The only thing that might slow that down is if the volume of food is so large that it gets stuck and requires a bit of extra muscular activity to send it down, and maybe a little bit more mucus application to soften it up a little bit. We've all probably experienced that at one time or another, where we literally bit off a little more than we could chew. And so it got stuck partway down, and it can be painful and uncomfortable. And a lot of that discomfort comes from the involuntary contractions in the interest of trying to push that food along the length of the esophagus to get it into the stomach. Okay, so if I can't remember what our color code was, but let's say our esophagus was blue and our stomach is green. As we approach as the food, we had it designated as the food is being passed along the length of the esophagus, there's not a very strong, but nonetheless a sphincter. We call that the lower esophageal sphincter. LES. And it's located right at the junction between the esophagus and the stomach. And keep in mind that this esophagus has traveled through the thoracic cavity. That 20 to 25 centimeters of length, by the way, I'm never going to ask you the question, how long is the esophagus? Dumb question, not necessary for us to spend time on. But the point is that 20 to 25 centimeters represents the distance essentially through the thoracic cavity. It's going to travel behind the trachea and then behind the bronchial tree to a hole that's in the diaphragm that allows it to pass into the abdominal cavity. And that's the junction where the esophagus will enter the stomach. The stomach contents typically are going to be below the diaphragm and the esophagus is above. And that's going to help with delivery because the delivery and prevention of regurgitation, this sphincter is designed mostly to prevent backup or backflow into the esophagus. As we've mentioned already, the contents of the stomach are going to be very acidic. If the acid is exposed to the esophagus, it's not equipped to respond or resist that acid, so it's going to cause damage. And most of you, if you've ever heard of or experienced heartburn, heartburn is actually gastroesophageal reflux, where the acid is now in the esophagus and you've got pain receptors against that acidity that tell you that there's something not quite right in that particular area. And because it's close to the location of the heart's going to sit right above the stomach, essentially, off to one side a little bit, but is right in that same location. So that's where the name heartburn came from. It's not actually got anything to do with the heart in that case. So we've got a lower esophageal sphincter that prevents backup. And we've got the small intestine also a sphincter at the small intestine side. The small intestine is going to be what the stomach empties into. And the small intestine we'll be talking about next after we get through a discussion on the stomach. So the stomach we can see is a little bit of a J-shaped structure. You can see that it's an expansion from the esophagus, and that expansion reflects part of its function. Part of its function, when we talk about the stomach, is that it's a reservoir. So the expansion reflects its role as a reservoir. It's going to receive volumes of food that once it starts mixing up with the juices or the secretions from the stomach, we refer to as chyme. Reservoir for food that becomes chyme. It's just the descriptive term of the mix of food, proteins, carbs, and lipids that have mixed now with the stomach contents, with the secretions from the stomach. So this stomach provides an expansion, a reservoir for a volume of food. Several liters of food and juice can be housed in this stomach. And one of the features of the stomach that makes this possible is that if we looked at the inside lining of the stomach, we see these very distinctive mucosal folds. These mucosal folds have a name. I'll write it out for you in a second. Those mucosal folds are called rugae. So this is a distinct feature of the stomach lining. Those folds allow for expansion of the lumen of the stomach. As the stomach fills, these folds become stretched. But because those folds allowed for stretching on the inside lining, it doesn't have the same impact on the outside lining. So the stomach itself won't distend or receive sort of tension until those rugae have been lengthened out because of the mass of food. So eventually you will feel some stomach distension. That's when you can sit back after your turkey dinner at Thanksgiving and say, "Woof, I feel pretty full right now," and pat that belly. But that requires a relatively large volume before you're going to be able to feel that, because these rugae allow for some expansion internally before you're going to feel it externally. So it increases surface area and increases the ability of this stomach to be able to act as a reservoir. So that's a major function. And this is one of the features that allows for that major function. And another major function of the stomach is that it is described as being a mixer. We're going to take the contents of food and mix it at the stomach. And when we mix the food at the stomach to be an effective mixer, we're going to see another additional feature to this organization. Remember, we said that the general arrangement of mucosa, submucosa, muscularis, and serosa allowed for 2 smooth muscle layers. But this tissue, the stomach, is a mixer. So we're going to see 3 smooth muscle layers. Which reflects its ability to mix. So we're going to have a circular layer, which was typical. We'll have a longitudinal layer, which was typical. And we're going to have an oblique layer. So if we consider the stomach, the tube of the stomach this way, we would have a circular layer. And we know the circular layer will affect the lumen, the diameter of the lumen. When it contracts, it'll squeeze closed, and relax, it'll dilate open. We're going to have a longitudinal layer that will shorten the length of that segment of the tube. And then we're going to have an oblique layer that, in the case of the stomach, is going to create a little bit of torque, a little bit of a twist. And the result of those 3 muscle layers being able to sequentially contract and relax is going to give us a much better ability to mix the contents that are in that stomach. Everybody okay with that? Does that make sense?
Yep.
Can you repeat that one more time, actually?
Yep. If we have three different layers, organization of sheets, smooth muscle typically is arranged in sheets. And we saw that in the heart. Cardiovascular muscle tissue is essentially smooth muscle that's very specialized in that location. But what was similar was that the cardiac muscle tissue was a sheet that created sequential contractions, right? We had those intercalated discs that gave us even more sequential contractions. But when we've got smooth muscle that's arranged circularly, it means that when that muscle contracts, we're going to reduce the diameter of, in this case, the stomach. And when it relaxes, we can increase the diameter. So that's going to create some mixing. When we shorten the length of the muscle in segments of the stomach, that's going to create some mixing. And then when the oblique angles contract, we're going to create a bit of a torque or twist in the muscle walls. So that's also then going to create mixing. So between the three muscle layers, we get more mixing going on than we would if there were just the two muscle layers. Does that make sense?
Yes, thank you.
Okay. So the other thing to consider then, from a functional point of view for the stomach, is that it's responsible for delivering small amounts of the chyme that's being produced now to the small intestine. So if we look at this stomach, the main part of the stomach, sort of this region right in here, is referred to typically as the body. The part that narrows towards the small intestine is the pylorus. And the part that's closest to the heart is called the fundus. So we're going to see that some of the cells and glands responsible for the chemical digestion that's going to go on in the stomach are arranged slightly differently depending where we are in the stomach. So there's not too much glandular activity up closest to the heart. That's mostly going to be about delivery. And we're going to start to see a change in that anatomy, the change in that arrangement of simple columnar cells as we move through the body and especially towards the pylorus. We'll talk about that in just a minute. But this valve right here is called a pyloric valve or pyloric sphincter. And that's what's going to regulate small amounts of chyme being introduced to the small intestine. So we don't want large volumes being introduced to the small intestine because we want to be able to digest and absorb, and that takes time. So we're going to introduce small amounts into the small intestine so there's more time and more space available for enzyme action in the small intestine. So that sphincter will open and allow small amounts of chyme to enter the small intestine and then close again. And it will be under the influence of chemical messengers. Everybody's pretty comfortable with that concept, I think, by now. So there are receptors there for hormones with names like secretin and cholecystokinin, and they will cholecystokinin in particular will slow down stomach emptying. So in other words, it'll cause the pyloric valve to close. So we'll talk a little bit more about that when we get to the small intestine. But here we're looking at the major responsibilities for the stomach. So the goal of mixing is to expose food to the gastric secretions. So you can appreciate that if we're mixing, because of these three layers of muscle, that would be mechanical digestion. But when we expose the food to gastric secretions, our goal there is to introduce chemical digestion, enzymes that are part of this gastric juice or gastric secretions that are going to help us with the digestive process. So when we look at gastric secretions, Most of us are generally aware that the pH of gastric secretions is very low. It might be somewhere between, well, let's say physiologically between 2 and 6 pH. There's quite a range there. It's more likely to be plus or minus sitting in that 3 to 4 range. So very acidic. And when it gets into other areas than the stomach, whether we're talking about reflux into the esophagus or delivery to the duodenum, those tissues are not equipped to deal with that degree of acidity. So there has to be mechanisms available to us that prevent that exposure. That's one of the roles for the lower esophageal sphincter. It's one of the reasons why we control the amount of chyme that's going to be introduced to the duodenum. So the primary acid that creates the gastric acidity is hydrochloric acid. And we have a cell, a parietal cell, that produces hydrochloric acid. And the parietal cell has several receptors associated with it that are going to regulate the degree of hydrochloric acid being secreted. So we have a parietal cell that's going to be influenced in an encouraging way by an ECL cell, enterochromaffin like cell. That's where the L comes from here. And entero usually is indicating the small intestine. That's why this cell is found in the stomach, but it looks a little bit like a similar cell that was originally identified in the small intestine. So this is a similar cell, but in a different location. So that's where the terminology came from. And it produces histamine. So we know that histamine is going to encourage the parietal cell to produce hydrochloric acid. We also know that ACH is going to have a stimulatory effect on the parietal cell to produce hydrochloric acid. Where would the ACH come from? What do those initials stand for?
Parasympathetic nervous system.
What's the neurotransmitter that the parasympathetic nervous system utilizes?
Acetylcholine.
There we go, acetylcholine. So, and we know parasympathetic promotes motility and secretion. So here's where some of that signaling is coming from. The vagus nerve releases ACH and targets the parietal cells and is stimulatory for the production of HCl. Another cell that we find, so this is another cell type that we find in the stomach that's going to promote parietal cell functions in the production of HCl is gastrin from a G cell. Nice name for a cell that produces gastrin, the G cell. So the cell is the G cell. It produces gastrin. Maybe what I should do is put gastrin up here. And then we can move our G cell in beside the number so that we're staying consistent there. So those are 3 examples of stimulatory factors on the parietal cell to produce HCl. So all of those would increase the production of HCl. Now we also have an inhibitory factor that's going to help us regulate, sort of keep in balance, check and balance, negative feedback if you like. And that's going to be coming from a cell that we call a delta cell. And the delta cell produces something called somatostatin. Statin stops, and somato tells you its body. So it's a hormone that generally is inhibitory on many metabolic actions and functions in the body. There's delta cells in the pancreas that we mentioned when we talked about the pancreas. We had alpha cells produced glucagon, beta cells produced what?
Insulin.
Yeah, insulin, good. And the delta cell produced somatostatin. So somatostatin has an inhibitory act on both the beta cells and the alpha cells with respect to blood glucose management. And here we see it's got an inhibitory effect on the parietal cell for the production of HCl. So that's another cell type. 4 cell types. We know there's a 5th cell type, the mucosal cells, and those mucosal cells are really important. Oh, can you see that okay? I can't. Our mucosal cells, guess what? Produce mucus. And the mucus is really important because as we produce these acids we know are going to denature proteins. So they would denature the proteins in cells. If the acids are exposed directly to cells, they're going to start to break them down. And that's the purpose from a digestive perspective of HCl, is to break down proteins. But we also have proteins in our cell membranes. So if we didn't have mucus lining the stomach wall, these acids would start to digest the stomach. So it's a pretty good mechanism that Mother Nature's figured out to prevent that from happening. Mucosal cells are going to produce mucus. So we're aware of that cell type as well. That's going to help protect us from damage from that acid. And a number 6, so that was a number 5, a 6th cell that we need to be aware of is called a chief cell. And the chief cell produces something called pepsinogen. And you know something about pepsinogen right away. What do you know about pepsinogen when you look at that word?
It needs to be activated.
That's right, it's not active. And you can probably guess what it's going to be called when it's active.
Pepsin?
Yep. And pepsinogen needs to be activated by the acids produced by these other cells. So when we're in a low pH environment, the pepsinogen that's being produced by this chief cell is going to be converted to pepsin. And together, HCl plus pepsin are going to break down proteins with a goal of achieving their building blocks, amino acids, sometimes small peptides. And amino acids, but we're taking big molecules and making them smaller. That's the point. So in the stomach, we see the mixing illustrating mechanical breakdown of carbs, lipids, and proteins. And we see the low pH environment and the pepsin specifically representing chemical breakdown of proteins. Now, these chief cells also produce something called gastric lipase. So again, we talked about that word lipase. The gastric lipase plus the lingual lipase are going to work on breakdown of lipids. So we get a little bit of chemical action on lipids in the stomach as well. I say a little bit because these gastric lipases and the lingual lipases, they're relatively small amounts working on lipids, but don't have a huge effect. Most of the breakdown of lipids and carbohydrates from a chemical perspective are going to go on in the small intestine. But we need to be aware that these enzymes exist here, so they do begin the process of chemical digestion in the stomach. So we have these distinct cell types that we need to be aware of that we find in the stomach mucosa and submucosa. So they're going to look like this. So here's what we call a gastric pit. And all the way along that length, so here's our stomach. Remember the J-shaped stomach. And the mucosal folds, submucosal, mucosal folds called rugae. And especially as we get down towards the pylorus, but through the body in the pylorus, if we look closer at the submucosal arrangement of simple columnar epithelium, we would see gastric pits. And the gastric pits are filled with distinct cell types that are going to contribute to the production of our hydrochloric acid. So here's our parietal cell. And we have some endocrine cells, the G cells that produced gastrin. And we knew it had a stimulatory effect. The D cells that produced somatostatin. And it had an inhibitory effect. Right? And some of these other cell types, we got some mucus cells, and up on the top here, as we get back into the closer to the lumen of the stomach, so just so you're oriented here, here's our lumen. We're predominantly dealing with mucus. So the mucus is going to line the stomach cavity as the acid is being introduced to that stomach cavity. And so there's a protective function. And what happens, for example, in ulcers is that this arrangement between the balance of acid and mucus is disrupted, and it allows bacteria and inflammatory mechanisms to access the lumen of the epithelium of the stomach and start to erode it. And that's what becomes an ulcer. So it's a disturbance between the balance of mucus production and acid production. And it results in too much acid relative to the mucus, and so damage is going to occur. So we would have, amongst these different color cells, we would also have our chief cells producing pepsinogen. And we named, so that was one of our cells, another cell. Oh, there's our chief cells down there. 4. Mucus cell was 5. And we named one other cell. What was the other cell we named? It was our ECL cell. And the ECL cell was the one that produced histamine. ECL was responsible for producing histamine. So we like histamine, ACH, and gastrin because they stimulate parietal cells to make hydrochloric acid, and the somatostatin inhibits that. So we get a regulatory factor in there. We get a factor that can balance the production of hydrochloric acid based on how effective the somatostatin is, how much is being produced by those delta cells in the stomach to help slow down that production. So most of these cells have more than one function. We sort of learned that in AMP100 when we did a project on a particular cell. We recognized that they do more than one thing. So our gastrin cells, our G cells, produced the gastric lipase. The parietal cells also produce what's called the intrinsic factor. Now, has anybody heard of the intrinsic factor before? You can say no if you haven't.
No.
Was it for blood clotting?
Not blood clotting, but you're close. It has something to do with blood cell production. So the intrinsic factor is an essential factor that promotes and allows for the absorption of vitamin B12. And if we don't have the intrinsic factor, we can't absorb B12. If we can't absorb B12, then we get dysfunctional red blood cells. An absence of B12 is going to create a blood anemia. And an anemia is a general term for any time our blood cells can't deliver enough oxygen to all the different cells in the body. So B12 is essential for the normal development of red blood cells and their ability to carry oxygen to all the different cells in the body. So this intrinsic factor is produced by parietal cells, and promotes the absorption of B12 so that we can carry oxygen. An absence creates a B12 anemia. It's got a fancy name, pernicious anemia, but basically it's an absence of the intrinsic factor. So occasionally, you may have heard of this, some people go in for surgery because they're dealing with weight issues, and they get what's called a lap band surgery. And what that does is it reduces the size of the stomach. So now the stomach volume is going to be, here instead of all the way through. But when you close off and block a part of the stomach, you're going to lose the function from the cells in that part of the stomach. One of the side effects of people that get this surgery, and it doesn't happen in all of them, but it does happen in a significant number, enough that it's been recognized, is that when you block off the function of a section of the stomach, you're in danger of reducing the production of the intrinsic factor, because in the interest of reducing the volume of the stomach, you've blocked function of the cells in that section that you blocked off. And so a side effect of some people who get this surgery is they end up establishing an anemia, a B12 deficiency that causes anemia. So that's you solved one problem, or attempted to solve one problem. There's lots of issues with that type of surgery, but you've created a new one, right? And that's not uncommon when we try to treat different types of problems in the body. We recognize multiple factors that are going to be affected. You can appreciate that if histamine stimulates parietal cell functions, what's a medication that a lot of us have taken at different times in our lives? An antihistamine. So antihistamines might be effective at dealing with an allergic reaction or an allergic response, for example. But we can appreciate that it's also going to have an effect on the digestive system, because if it's an antihistamine that we ingest through medication or inhalers or whatever the case might be, it's going to affect the role for histamine all over the body. And in this case, it would reduce the production of hydrochloric acid and may create digestive problems. Do you follow me on that? Does that make sense?
Yeah.
Yeah. Okay. So another diagram I'm going to show you that helps us understand the interactions between all these different cells in the stomach is this one. So here we have our stomach. So there's our fundus, our body. This would be the pyloric region down here. And we're showing the cells that would be found here. So here's our vagus nerve. Oh. So what's going to come from the vagus nerve? ACH. So an ACH acts on the ECL cell. You see it's got a positive effect on ECL, and that makes sense, stimulatory. So it produces histamine. And the histamine, we've got receptors on our parietal cells for histamine and for ACH. So both of those would be stimulatory. See the check? Stimulatory. Here we've got a somatostatin receptor, and the somatostatin is coming from D cells at the antrum, or the one part of the stomach, and another part of the stomach. D cells are releasing somatostatin into the circulation, and somatostatin will have an inhibitory effect on the ECL, and it has an inhibitory effect on the parietal cell. So this is a diagram showing everything that we've just been talking about. There's our G cell producing gastrin. Gastrin's a hormone, so it's going to go through the circulatory system, and it's going to be excitatory at this receptor. The cholecystokinin receptor is also going to recognize cholecystokinin and gastrin. So it's a receptor that can recognize both. In the stomach, it's going to be about this gastrin in this case for the production of hydrochloric acid. Do we see this okay with these cells here? Our D cell is otherwise known as our delta cell. So it's going to be inhibited. The delta cell is actually inhibited by ACH, and that makes sense because the delta cell makes somatostatin. The vagus nerve is interested in encouraging secretions from the stomach, so it's going to do everything it can to stimulate So it stimulates the ECL cell. It's going to stimulate the parietal cell, but it's going to inhibit the delta cell. We see that okay?
Yep.
Okay, so antrum is also the pyloric area. See, they call it, in this case, they call it the antrum, but it's the pyloric area. It's the area that's narrowing just before we get to the small intestine. So all we're saying here is down at the antrum areas where we see more of the G cells. The gastrin is going to be in higher concentrations towards that end of the stomach. That's the only reason that these two areas were separated or designated as being separate. So when we looked at our stomach then, we had our stomach, I think we had it. So there's our esophagus with a sphincter. And our stomach, which was a reservoir, so it was an expansion. Rugae. And gastric pits. Those are the big sort of things to be able to recognize. The gastric pits increase in number as we get closer to the small intestine. And we're going to mix the food, create chyme, expose it to hydrochloric acid and to pepsin, and to a lesser degree, the gastric lipase, so that we get mechanical digestion of all the food groups because of the mixing. And we get chemical digestion mostly on proteins through the hydrochloric acid and the pepsin, and to a lesser degree on the lipids in the stomach through the lipases that are activated in the low pH environment. When we enter the small intestine, we're going to be regulated, that entry is going to be regulated by the pyloric sphincter, and the pyloric sphincter is going to allow small amounts of chyme to enter the small intestine. So that's the next tissue that we want to talk about. And the small intestine is only small in diameter. That's why it's designated as a small intestine. By length, it's really big. So when we're looking at the small intestine, we're going to see some changes here as well. So the general organization holds true, but with some specialized features that reflect functions at this level of the digestive tube. Small intestine is somewhere between 3 and 5 meters in length. So in a living tissue, it'll be somewhere between 3 and 5 meters in length. In death, this tube loses its elasticity. So if you stretched it out in death, it might be as long as 20 or 22 feet long. In life, because of the elasticity and its tendency to want to stay coiled up within the abdominal cavity, it's shorter. It's 3 to 5 meters in length, which would be about 15 feet or so, coiled up. But the small intestine is divided up into 3 distinct segments. The first segment, the one that is exposed to the chyme, is called the duodenum. It's the shortest segment of the small intestine. And then the middle section is called the jejunum. And the last section is called the ileum. Notice the spelling of ileum, I-L-E-U-M, right? We talked about an ileum, I-L-I-U-M. In the skeletal system, it was one of the bones that contributed to the pelvis. So spelling becomes important here, because if you spelled it incorrectly, you could be talking about a whole different system. So the duodenum is about 10 or 12 sort of centimeters in length. It's going to receive the contents from the stomach. The one thing we know about the stomach is that it is very acidic. So the duodenum is going to have some protective mechanisms associated with it, with that understanding that we're going to be exposing it to acids to begin with. So when we look at this small intestine, we're going to see that there's some specialized features. One of those is going to have to be what happens to neutralize some of those acids. The other thing that we're going to notice is that we're going to see a huge number of factors that are going to be responsible for promoting surface area. We're going to see some features that increase surface area because once we get to the small intestine, we have to finish digestion and we have to introduce and complete most of absorption. So in order for us to do that effectively, we're going to need some surface area. We're going to need to increase surface area so we create more exposure to enzymes and more and longer exposure to enzymes. And that's partly why we see this coiled tube. It twists and turns in the abdominal cavity that allow about 5 meters of coiled tube to be located in that abdominal cavity. So that's part of the surface area feature, is that coiled tube. And then we're going to see some additional features. We're going to see some features on that columnar epithelium. So the columnar epithelium, simple columnar epithelium, is going to form finger-like extensions. So these finger-like extensions can come up and we'll see these finger-like extensions of our columnar epithelium. And on the surface closest to the lumen, we'll see these additional extensions of the cell membranes. So this extension of the cells themselves is called a villus or a villi, and the extensions on the cell membranes of those villi are microvilli. So that increased surface area is referred to or is named as a brush border, because under an electron microscope, it starts to look like the bristles from a brush. So they describe it as a brush border. But what it does is it increases surface area. And you can appreciate that if we have enzymes in the location, these enzymes can be located in and around all of these microvilli and villi to create more exposure and more time for these enzymes to be exposed to the food. So that's an important feature that we're going to talk about throughout this small intestine. So most of the digestion that started in the mouth and in the stomach has to be completed in the small intestine if we're going to be able to absorb within that small intestine. So in addition, one of the features that we're going to see in the small intestine are Brunner's glands. So Brunner's glands are concentrations of cells that produce mucus. And a big component of this mucus would be bicarbonate. And bicarbonate is going to be able to neutralize some of the acids that come from the stomach. So these are the glands that are going to be responsible for neutralizing some of the acids that come from the stomach. So the Brunner's glands are located really sort of throughout the small intestine, but concentrated in the duodenum. So they can decrease pH from the stomach. So that's one of the features that we describe as unique to the small intestine, and in particular, the duodenum. So we're going to be able to produce mucus that's alkaline to neutralize the acids from the stomach. It also provides an alkaline environment for activating. So we're going to neutralize gastric pH and activate enzymes from the pancreas, which we'll talk about when we get to the pancreas and its contributions to digestion. So Brunner's glands play a number of roles in that respect. And again, the mucus is going to lubricate these sections of the small intestine as well, which is what mucus cells are essentially responsible for throughout the digestive system. So the other thing to recognize about the duodenum is that it receives chyme from the stomach. It receives pancreatic secretions, which are going to be chock full of enzymes that are going to help us with the chemical digestion, the final steps of chemical digestion. And it also receives bile from the gallbladder. And we'll talk more about that a little bit later on. So bile, we're going to find out, is essential contributor to lipid digestion. And we'll talk a little bit more about that as we get there. So the enzymes that are going to be received from the pancreatic secretions are released from the pancreas inactive and are going to become active when they're exposed to a more basic environment, which is the responsibility of those Brunner's glands. And again, the chyme is being introduced. It's acidic, so we're going to neutralize that. Then we're going to activate some of these enzymes, and we're going to be able to complete the digestive process in the duodenum and enter into the jejunum. So it's a continuous tube. So there's going to be a gradual conversion from digestion to absorption as we move along this small intestine. So when we look at the villus, the finger-like extension there, it has a network of capillaries that are going to make it available. So if we looked at the villus, this finger-like extension would be up like this, and our cells would be arranged in a simple columnar arrangement like that. And in particular, when we get up into this loop towards the lumen, these columnar cells are going to have those microvilli extensions on them to create that brush border that we talked about. And inside this villus, we're going to have a capillary that delivers oxygen and nutrients to the cells, and a vein that's going to take waste away. So these vessels are going to be down here in the submucosal areas of the wall of the intestine. And in the center, we're going to have another vessel. This green vessel that I'm drawing in here is going to be represented by what system?
Lymphatic.
That's right. Yep. Thank you. So we got an artery, we got a vein, and this part of the lymphatic system we called, when it's picking up lipids from the digestive system, we called that vessel a lacteal. But it's basically a lymphatic capillary, and it's going to be responsible. Remember, we said one of the roles of the lymphatic system is to pick up lipid molecules, larger lipid molecules that couldn't be traveling in the veins, instead are going to travel in a lacteal. And we know the lymphatic system eventually returns its lymph to the cardiovascular system in those thoracic ducts before it gets to the right side of the heart. So we know that these lipids that are being carried in the lacteals ultimately are going to be introduced to the circulatory system. So in the small intestine, particularly where we're going to see absorption occurring, the microvilli are going to be coated with enzymes. And these enzymes, as the food particles are being introduced to the small intestine, particularly the jejunum, plus or minus 90% of absorption occurs here in the jejunum. So these microvilli and villi and another feature that I haven't mentioned yet that I'm going to show you in a picture, increase surface areas so that we've got lots of time for exposure to enzymes. So some of these enzymes here that would be found in the brush border are going to have names like sucrase. We know that sucrase acts on what?
Sucrose.
Yeah, sucrose. And we know sucrose was a disaccharide made from glucose and
Fructose.
fructose. So the enzyme sucrase will break down sucrose to the monosaccharides, glucose and fructose. Now, as a monosaccharide, they can be absorbed. And we're going to see they'll get absorbed and they're going to be introduced to the veins. And we'll see why in a few more minutes. To the veins and carried away from the small intestine in the veins that originate here in this villus. So we might see an enzyme here also called lactase on lactose, that's going to break us down to the monosaccharides, glucose and galactose. And again, they'll be absorbed into the venous system and carried away from the digestive tract. Maltose maltase acts on maltose to break us down to glucose and glucose, monosaccharides that are absorbed. We're going to have lipases that break lipids down to fatty acids. Fatty acids can be absorbed by these cells, and sometimes what happens inside those absorptive cells of the digestive system is that the fatty acids are resynthesized into a molecule called a chylomicron. We'll talk more about that when we do the metabolism part of this digestive system, which is not this week. We'll do that next week after the test. We'll at least get it started. So these chylomicrons are triglycerides and some cholesterol and some protein molecules. They're too big to be delivered to the circulatory system, so that's what the lacteal picks up. And the chylomicron, just to plant this in your brains, chylomicrons are big molecules. They're going to leave the absorptive cell through exocytosis and then be picked up by the lacteal to travel in the lymphatic system before they're reintroduced to the circulatory system. Okay, but they're still being absorbed as far as that goes. So the concept is still the same, absorption here at the jejunum, but in the case of chylomicrons, they're going to be picked up by the lacteal. The monosaccharides and fatty acids, they can be picked up by veins. They're smaller, so they can be picked up by veins. Same with amino acids. They're going to be picked up by the veins and carried away. So that represents about 90% of absorption. The other 10%, a little bit of absorption of some electrolytes and water can occur in the stomach, and a little bit more electrolytes and water can be absorbed in the large intestine, just in the interest of making adjustments to homeostasis for those particular elements, water and electrolytes. So when we get to the jejunum, these villi and the other feature, so we'll write down villi. I'm going to show you what that looks like. And the feature of circular folds. We haven't talked about that, but that's another feature that we see, particularly in the jejunum, but throughout the small intestine are these circular folds. So these are more mucosal, submucosal folds, not the individual cells creating these villi. Let's take a look at what that looks like, and we'll come back to this slide in a minute. That slide is... Here we go. So when we look at this slide, here are the muscle layers. So we're good with the muscle layers, with the circular and longitudinal, just like we expect to see. But now we're looking at the circular folds. We can see these circular folds. So these are circular folds that are created by the submucosa and mucosal membranes, creating sort of these folds within the orientation of the lumen. So they create surface area. Then we're looking at villi, these extensions here. So these are whole cells that are created by these villi. So here's a single villus and our simple columnar epithelium around that villus. And in this larger picture over here, we can see the microvilli. So you can appreciate surface area. Thousand-fold increase in surface area because of this combination of circular folds, villi, and microvilli. And here we see the lacteal, nice pretty green lacteal, going to carry away those chylomicrons. And the venous system is what's going to carry away those nutrients that were absorbed through these cells and then picked up by the circulatory system. So the artery is supplying oxygen and nutrients to the cells, and the absorptive cell's function is to take nutrients from the lumen, they would be located up in here, and absorbed across those cells and introduced to the circulatory system and carried away. We okay with that?
Yep.
So if we come back down and take a look here, so the ileum then, so the jejunum is going to be the location for most of the absorption that's going to occur. And the other feature that we see in the jejunum, and this is the way that the smooth muscle works. Remember, we talked about peristalsis. Peristalsis was always about propelling the food down the length of the tube. So we're going to go from the esophagus to the stomach, from the stomach to the small intestine. And we know from the small intestine, we have to get to the large intestine. And in order for us to buy time in the small intestine to get the finishing up of digestion and 90% of the absorption to occur, we're going to also see a process that sort of slows down peristalsis. And that's called segmentation. So segmentation is a unique feature to this, primarily the jejunum of the small intestine, where we get alternating contractions and relaxation of the smooth muscle that creates a back and forth of the contents in the small intestine, back and forth movement of chyme. So our bolus of food is going to be propelled downward, peristalsis, and then kind of stops and is driven backwards, and then forwards and backwards and forwards. That's what segmentation is referring to. And that's a contraction cycle that's unique to the small intestine, in particular, the jejunum. So this segmentation is a type of mechanical digestion that is allowing more exposure to enzymes, chemical digestion, right? Again, we're combining our chemical and mechanical digestion components. We should all be comfortable with the idea that these mechanisms would be cooperating with one another with a common goal. In this case, the common goal is buying time for exposure to enzymes to get our molecules small enough that they can be absorbed by the cells responsible for absorption. So our small intestine is going to have mechanical digestion, which is our segmentation and peristalsis. And that segmentation is localized contractions that's going to allow us to mix chyme back and forth, kind of like if we want to go back to our toothpaste tube, if we squeeze in the middle and then squeeze from the ends, and then go back to the middle and squeeze, and then go back to the ends and squeeze, that's what segmentation is, right? Not like what we saw in the heart. When we squeezed from the bottom of the tube to create a sequential contraction towards the valves in the heart that gave us one-way flow for blood. In this case, we want back and forth. We want mix. So we're going to get squeezing from the middle and then squeezing from the ends. And that's going to slow down peristalsis. So food that enters the stomach might leave the stomach within a few minutes, usually more like 30 or 40 minutes. The first amounts of food can be mixed with the acids and then delivered to the small intestine. But in the small intestine, the food's going to stay in there for hours. And one of the reasons is the coiled arrangement of the small intestine. And another one is segmentation. And then we've got our surface area for exposure. So all these features represent structural and functional differences to the tube arrangement. Remember, mucosa, submucosa, muscularis, and serosa. So we've got specialized features here that represent the functions we expect to see in this area. Now, to squeeze in the ileum, the last part of the small intestine. So what we've got at the ileum is the presence of some Peyer's patches. We talked about Peyer's patches, indirectly at least, when we talked about the immune system. The Peyer's patches are an example of a mucosal associated lymphoid tissue. In other words, what we're going to see here at the ileum is a large concentration of lymphocytes, white blood cells, including some T cells and helper cells and so on, that are going to give us an immune ability for some recognition. We're going to be able to have some lymphocytes available to us here for the recognition and introduction, in other words, activation of white blood cells, lymphocytes, on exposure to bacteria and or viruses. So remember, we said when we started our food ingestion, we introduced a bunch of bacteria. So if there's still some bacteria that are going to be recognized, we've got some ability to recognize through these mucosal associated lymphoid tissues. That was the acronym for these Peyer's patches. So we got that located in the ileum. And then we have what's called the ileocecal valve. So it's an ileocecal. That's supposed to be a C there. Ileocecal valve. And that valve is going to regulate the contents that are found in the end of the small intestine into the large intestine. And that will look a little bit like this. So the ileum is here, just to orient yourself, and our ileocecal valve is emptying into the large intestine. You can see that the large intestine has got a cecum, which is the blind end of the large intestine, and on the end of that is our appendix. Our appendix is, we know the appendix is filled also with lymphocytes and other immune cells, so we don't really understand a lot about what goes on in the appendix, and I guess in the grand scheme of things, it's not significant, but occasionally it does get inflamed, and when it gets inflamed, it can be very painful, and it does increase the risk of rupture. And if it ruptures, it allows those bacteria that caused the inflammation in the first place to escape, and that can become life-threatening. So when it gets inflamed and causes symptoms, it's often removed. But as far as we're concerned, the content of food that was not absorbed in the small intestine is going to be introduced to the, let's go here, large intestine through the ileocecal valve. And that mass of now stool or feces is going to accumulate and be driven up the ascending colon, across the transverse colon, and down the descending colon, enters the rectum. Sometimes this S-shaped component here is described as the sigmoid colon. It's part of the descending colon, and enters that mass of waste is going to enter the rectum. And when enough mass enters the rectal walls will stretch, and that will initiate what we call the defecation reflex. The defecation reflex is partly autonomic and partly somatic. So if it's autonomic, we know that it's subconscious, and if it's somatic, we know that it's conscious. So the autonomic reflex is in response to stretch. So stretch is literally going to send a signal to the spinal cord and back out again to cause, so the stretch stimulus creates a spinal reflex that causes rectal smooth muscle to contract and the internal anal sphincter to relax. So the internal sphincter is not labeled here, but the internal sphincter is literally internal to what's labeled here as the external sphincter. So when the rectal smooth muscle and the internal anal sphincter, when the rectal smooth muscle contracts and the anal sphincter relaxes, that's going to allow elimination to occur. Up until the age of 2 or 3 or 4 years of age, this is the only component of the defecation reflex that's active. As we develop our skeletal muscular system, we add in the somatic component, and the somatic component signals the external anal sphincter. And the external anal sphincter is skeletal muscle, so it's under voluntary control. And when we feel that stretch and initiate the autonomic reflex that would allow the excretion of this waste, we can overcome it with conscious signaling that activates the external anal sphincter to constrict or contract. And if the external sphincter contracts, it prevents the elimination of the contents from the rectum. So typically what will happen is the rectum will relax again, at least for a little while, but as it starts to fill further, a new stretch reflex will be initiated and a new spinal reflex will be generated that causes the rectal smooth muscle to contract, the internal anal sphincter to relax. And if we don't have the somatic response, we get elimination. So really, the learning of the somatic response gives us the option to choose when to defecate, when to eliminate waste from the digestive system. So that's what our defecation reflex is, a combination of autonomic and somatic. The somatic part is learned. It comes with maturation of the skeletal muscular system with respect to that external anal sphincter. We're going to see the exact same concept when we look at the bladder, when we look at the renal system and the urinary system, and the bladder has the same concept. When the bladder stretches, there's an internal sphincter that will relax, you'll urinate. But there's an external sphincter that we can control that will prevent that from happening within reason. If you continually try to overcome that stretch reflex, eventually you're going to lose. And eventually there's going to be release because that stretch will be too forceful. So what forces the stool or the waste from the small intestine through the large intestine are strips of longitudinal muscle. So these strips of longitudinal muscle are a unique feature that we see in the large intestine. These strips are called the Taenii coli. I know they got an extra A in there, but you'll see it spelled a couple of different ways, American versus British, et cetera, et cetera, et cetera. So these are strips of smooth muscle So rather than a sheet of longitudinal muscle, we see in the large intestine these strips. There's 2 or 3 of these strips along the length. And what they allow, smooth muscle, they allow mass peristalsis. And most of us are well aware of what mass peristalsis feels like. When you get cramping in the abdomen, either because maybe you're experiencing some constipation or maybe you're experiencing diarrhea, often there's a reason for that. It might be an infection, whatever the reason, you can feel very forceful contractions, very forceful cramping in the lower abdominal area. That's mass peristalsis. And that's a unique feature to the large intestine. Remember, I mentioned about 90% of the absorption that's going to occurs in the jejunum, but another smaller percentage, so a small percentage of absorption occurs here in the large intestine, mostly of water and electrolytes in the interest of balancing out homeostasis. And you can appreciate that if there's a larger component of water in the stool, it's going to be runny, and less percentage of water in the stool, it's going to be hard. Part of constipation is the stool has stayed in the large intestine for an extended period of time and has dehydrated. It gets hard to move. And in diarrhea, it's the reverse. Diarrhea is very runny because it's moved very fast through the large intestine and water hasn't had time to be absorbed. So those explain the consistency of the fecal matter, the feces, that is being excreted under those circumstances. One of the reasons that we like fiber in our diet is it's a carbohydrate that is indigestible. Literally, the definition of fiber is indigestible or undigestible carbohydrates. So the fiber from certain plants is, we don't have enzymes for it. So that type of carbohydrate is called cellulose. You know, it ends in OSE, you know it's a carbohydrate. You're pretty sure about it anyways. But we don't have an enzyme called cellulase like a cow would have, or other animals that feed off of plants primarily. So in this case, it's indigestible, so it's going to be contributing to the mass that's delivered to the large intestine. And it's very good at absorbing water. So it keeps the stool moist and bulky. Some people are, it's too early in the morning for this kind of conversation, but we like our stool to be bulky and moist because that's going to increase motility. So in other words, prevent constipation. So fiber in our diet has a number of advantages, and one of those is this one. Increases motility through the large intestine by keeping the stool moist and bulky. And so that's going to help with our motility and help with regularity of excretion, the removal of waste from the digestive space tract. So this area of the large intestine is, somewhere between 1 and 2 meters in length, as opposed to the small intestine that was 3 to 5 meters in length. So it's referred to as the large intestine because its diameter, it's much larger in diameter. We probably all recognize that this is a primary location for the normal microbiome of our digestive system. A large concentration of bacteria is located here, and it's part of our normal flora, normal biome that we expect to help in a symbiotic way, help us with functions. So for example, the high normal bacterial count in the large intestine improves our ability to absorb vitamin K. And we know vitamin K, somebody answered earlier what the role for B12 was. Now your answer is going to work. What do we like vitamin K for? It's directly involved with many of the steps associated with
Clotting.
blood clotting, right? So that's the vitamin you were probably thinking of when we were talking about B12, but this is vitamin K. Some of our B vitamins also are better absorbed with this exposure to bacteria in the large intestine. So we have a number of responsibilities here that are important for normal functioning going on in the body. So it isn't just about feces. It's not just about excretion. Lots of other roles that go on here. Okay, let's see, where are we? Okay, just before we take a break, we're going to take a break in just a second. What I want to do here is just look very quickly at our summary of where we would see mechanical and chemical digestion. Okay, so here we are in the mouth. We know mechanical digestion is going to be the result of chewing, mixing up saliva. The tongue can help us with the mixing part. The teeth help with chewing, and the voluntary muscles are going to initiate swallowing. Chemical digestion of carbohydrates is going to occur here. We're not going to. Okay, why is that not writing? Oh, maybe why? So we know that there's some lingual lipase release, but it's not active in the mouth. So we're going to eliminate that component there. Now the esophagus is all about delivery from the mouth to the stomach and initiates the involuntary movement peristalsis. And then once we get to the stomach, we're going to see mechanical digestion of anything, the contents of the stomach. Okay, remember, we're going to see rugae, the folds of the mucosa and submucosa within the stomach. Chemical digestion of proteins and fats are going to go on here, pepsin and hydrochloric acid, and our gastric and lingual lipases. Absorption of a little bit of lipid-soluble elements can occur here in the stomach, but again, a small percentage. Once we enter through the pylorus, or the pyloric valve, lower esophageal valve, pyloric valve, and then our ileocecal valve, so we're going to just highlight our valves here. Each segment is going to be separated by a valve. Mechanical digestion is mixing and propulsion. Chemical digestion of carbs, fats, proteins, nucleic acids occurring in the small intestine, and the absorption of these things. About 90% or so are going to occur in the small intestine. Then we move down to the large intestine, some segmentation. Our segmentation is really occurring mostly up here in the jejunum. Mixing and propulsion is going to occur and be delivered to the large intestine. No chemical digestion except a little bit by bacteria, absorption of some ions and water. And then rectum, if the rectum stretches, it's going to initiate removal. And we started with swallowing. We finished with defecation. And in between, we've digested, secreted, and absorbed. Okay, so for this segment, for what we've been talking about, I'm going to expect you to be able to relate to those 5 functions: ingestion, digestion, secretion, absorption, and excretion. And with respect to each of the individual segments, recognizing that if the segment has a different responsibility, it's going to have a different set of anatomical or structural features associated with it. So way back at the beginning, we color-coded that. And that color code gave us our indicator that we were going to have a different function if we had a different color. In other words, the color represented structure, and the different structure would reflect functions in those areas. So these are the things that you're going to want to be able to explain and discuss with respect to the esophagus, the stomach, the small intestine, and the large intestine. Okay, let's take a break. And